the recording will work. We'll see. So we saved the day. All right. So we got some red text moving on ahead, but we'll be all right. But fun, cool thing, because we're all advanced bio majors, but one of my favorite things learning from this is that at the end of the day, a living thing, not made of living things. The cell itself comes from non-alive amino acids, carbs, lipids, boring stuff. But a cell can do a lot of things. Our drugs are not alive. The only thing that we'll meet that's alive, a living drug at the end. That's it. We are at that stage, though. It's exciting. Okay, let's do this. All right, red text time, because yeah, it's a week-long class, and we're doing it. Here we go. Cells, in order to communicate, and do strictly think of it as a language that they speak between each other. Some of these signals, most of these signals will be contact like that. Cells will tell each other to turn off, on, die, make more, whatever it takes. All of this is happening in you right now. And it's these receptors right here. It's these interactions. The target of most drugs is to prevent this language from being spoken when it is being spoken too much. Or it is prevent, or it is to augment this language to be spoken. We need it more. Receptors are one way to do it. There are better ways, though, more final ways. You can be local. You can explode out with signal as a cell. Pieces of signal will be met by other cells, eaten up, and told what to do. That cell will react. Reaction, action, reaction. Most local signalings will be cell to cell, but not direct receptor contact. But in here, the action potential, everybody's favorite, as we just saw, there are little receptors waiting for these little guys. A lot of our drugs will stop this interaction from happening, or they will stop that neurotransmitter from being recycled in some cases. We'll see. So we can play with physiology, with chemistry. We can fight back with it. We can stop these signals. The long-distance signaling that we talked about with the endocrine cells, that one's tougher because we are trying to stop an entire world from communicating back and forth. But at the end of the day, you have plenty of cells, each with the same genome, but they decide how to use it. Some cells can receive the signal, others can't because they don't use that gene. Only certain cells secrete certain signals. Sorry for the little backup echo, we'll fix that later. But this language is where we intervene in this class. We stop this language, we correct the mistakes. At the end of the day, getting away from ideas of what's a nutrient versus a drug, things like that. We talk strictly about a drug. Let's go ahead and see it. Number one. Here, I'll even go vertical for us on this one. Can't be a drug if it doesn't do anything, right? Needs to treat and target something. It must intervene on a biology process of some sort. Is your number one. I'll give you little keywords to nash these notes down sometimes. It's one of the ways I try to select info for you. Moving on to number two. In a perfect world, you don't need the drug to go where it's not needed. Ideally, you design a drug that goes for the heart, if that's the target, and it doesn't bind to something in the brain. Number two's theme, I would say, is targeted. You have to actually do something. In a perfect world, you are doing that thing in the right place. Who's ever read all the side effects of drugs or seen the commercials? Yeah. Last little guy. Whatever your target is, this is one of the hardest parts. You need to be able to get a chemical to it. I'm going to say 3 is just the word right there. Excess. This is what you need. Call something a drug. Theoretically, we should be hitting these 3. We will find that things get complicated and that plenty of drugs work for their byproduct effects. For example, do you know Rogaine is a heart drug? Whoops. Works how it works, though. You can go in black right here if you want. True definition, it's fine. It's not something I'm going to strictly test you on. A good idea of what I would ask in an exam, some of you know me from this, is I could ask a question that would be more along the lines of like, what is one thing that a drug ideally would accomplish of the 3 primary? That's a good way to assess that with a smaller vein. Play to your strengths with that style of exam question as well. There'll be times for the mysteries, but hopefully this setup is pretty straightforward. Now, 3 is actually really hard. There are undruggable genes and targets. Where do they live? I'm not going to go to cards on this one. Yeah, there you go. There's a big restrictive, big border setup before that blood gets to the brain. A lot of filters happen right there. A lot of brain drugs have to be a little more direct to reach that border. Number 2, remember everybody loves cells, right? Yeah, we love cells. You still got to get in that nucleus to get a lot of those genes. A lot of the genes that we want to drug, they're all the way in the nucleus. Getting past 1 cell membrane is hard enough, but 2 and 1 that's really secure, that's tough. There's a lot of cancer genes out there. Can't get to them. Perfect. So let's take a look at caffeine. Literally like 3 of you on the card were like caffeine addict. This is what to remember me by. Come on, stop it. All right, basic mechanism of caffeine. There's a little signal spit out from some of your neurons called adenosine. It accumulates throughout the day in your circadian rhythm, and it stops you from moving more, settles you down. You get tired eventually, right? Not if you pop up Celsius, right? That caffeine's going to block the site of adenosine. No more tired. So, caffeine's complicated. There are more ways that it works. It does serve as a stimulant outside of this mechanism, but I wanted to give you a blue text example of 2 cells communicating and we cancel them out. Perfect. Ooh, it'll be fun. Who thinks that it's possible to actually become addicted to caffeine? I think I might be speaking for myself a bit, but hey, what are you going to do? So, our favorite thing besides chemistry, plants. Everybody loves plants. Come on. Violets love plants, I know that. All right. Most actual substances have their beginnings right here, even to this day. Typically, there is some sort of reaction and raw material in a plant. We can filter that down. We can do a bunch of fun analytical chemistry for this. Eventually, you can make it accessible to patients. That key part right here, we'll find next class. This is a hard thing to do. Getting in the form that can actually reach patients and not kill them, by the way, can be a little tough. Because yeah, you don't want to inject raw opium into anybody. They'll die. That'd be that. As the story goes, this is what we're after. And although human phys sometimes it felt very big and broad, the interactions were happening at this little level right here. Whether you're open, closed, or really open. A familiar character is the example in this case. Yeah, who was it? We talked in Cancer Bio when we did the podcast. I asked you guys, I was like, how many people actually vape? Like 15%? And what was it? I think Sarah was like, it's more like 45. And I was like, oh, that really came back for that one. So, what's trying to kill you? Pathophys. What's trying to stay normal? Phys. At the cell level, It's all part of the plan. A lot of you in this room, I believe, have already had cell as well, alongside human phys. You'll see it's a lot like that, because we will be zooming in on a lot of these. So, we want to kill the cells, though. Those receptors, things that are sticking out of the cells, that is typically going to be the target. They are the easiest access, right? Nicotine will access those nicotinic receptors. Easy access, like that number 3, right? Receptors are always up for grabs. They're our best drug targets. Stop them, make them turn on, whatever you want. So, to more broadly get at this, I haven't resized all my slides correctly. Sorry. I'd say give this a nice blue text right here, but hey. The goal, the complete goal of a signal to a receptor is something along these lines. Signal is received by a receptor, in this case, a growth factor. I'm going to say number 1, number 2, 3. The receptor often sticks inside the cell and outside. It will have partners. These will activate each other in a sort of avalanche. I would characterize this as a cascade. Those genes are not the target, true target, is the genes that they will activate that will turn on other genes, transcription factors. When you have sugar, when you take drugs, genes activate inside you. It is not a passive thing. Transcription factors will turn on new genes, and a new effect will happen. Now, in this example, this is likely cancer pathway. There's too many of this. There's too many of those, potentially. We can drug. As easiest to drug is that number 2 right there, that receptor. In a perfect world, it is just waiting for us to kill. Now, we'll meet characters next week that are more, we'll meet characters next time that are, some are more positive pushers, other are antagonists in the story. Some push, some stop. In a lot of cases, though, when you drug something, it usually blows up and dies. That's usually the case. There's plenty of little chemicals we've designed that keep things open for business. Now, an interesting little case study of this, one of my favorites. Who's ever heard of toxoplasmosis infection? This is read text. Cats get it. Humans can get it too. So what happens with this poor thing is that the tox, the little plasmosis, it gets into mice, and it mimics a signal, one of our neurotransmitters called GABA. GABA calms the other neurons. It says, stop, don't do anything. Without those neurons going, the mice lose all fear, and they walk out of the hole, and guess what's waiting there? Cat. And it eats them. Cat poops it out, mice eats the poop. Cycle of life. But even the smallest things can influence behavior, including humans. Higher risk-taking people were often significantly associated with, they had cats, and they were infected with tox. They had too much GABA. Signal calmed them down too much. They didn't have any fear response, or lowered. Very naughty. You can get it from raw meat too, so don't let your dogs be bad. And don't eat raw meat. I've seen that stuff from Wisconsin where they eat the raw ground beef. But yeah, we have natural signals, natural neurotransmitters. Infections can change this, but if infections can change it, what can we do? Influence all kinds of these pieces. CNS, central nervous system stuff will happen week 4. It's my favorite. It's one of my favorite parts of the class because you see how quickly the brain can change. I did see a bit of a comic, I suppose. I don't know if I'm going to be able to write this up here. It was basically like, here is what I thought, here. All right, you see that little patient right there? Here's, in the comic goes, here's what I thought anti-anxiety meds would do. They thought they were going to make a little smiling face, but the comic just basically said, here's what they actually did. You'll find that a lot of those meds are a bit rampant in what they do, and not everybody's genes are the same. Not everybody's receptors respond the exact same way. Remember alleles, dominant recessive, that's only two. Most of your brain receptors have hundreds of different alleles, different possibilities. So, looking pretty good. Go ahead and kind of collect yourself. Write one or two things that you're good on, write one or two things that's in the margins that I have a question about that. It's a good way to take a little break right here. We got a bit of chemistry coming. Don't worry, another break slide though. And again, if you have a random thought, you're like, hey, how does this work in class? Remember to put that on the back of that intro index card. Okay. Trying to do with this, write two things you're good on, write two things you have questions on. It's a cognitive technique that play your strengths. It's two things that you're very good at now. They are going to relate to what we do next, and they could relate to what you're not as good at. It can cement that, it can serve as anchors for that. Taking a moment to identify what's not going perfect right now. It's the perfect ammunition to study later. Find me to help clarify. One of those ways that you can take notes without just taking everything that's already written. I don't want you to do that. That's why slides are posted. All right. Who's ready for some good old chemistry? It was a prereq. Well, one was. We're gonna have such a good time. All right, here we go. Similar to those 1, 2, 3 that we talked about. But a good drug, a marketable drug that's not going to kill the patient, it's going to work, is what you need. It better have a target. It needs a place to land. Some drugs can bind multiple spots. That's fine. It needs to do something. It needs to hit something. It needs to grab something. I don't know. Number two, you better be able to do this multiple times in most people. There are plenty of drugs that only work for like 5% of people, but whatever. If they're not marketed well, and they don't usually show up because we'll find out in a bit. Money's an important part of this. Number three, find out that it's a little harder to get drugs into people helpfully sometimes. Taking Ozempic as an example. It's not a pill. It's a big old needle right there. Surprised people would trust us with that. Right there. That is something we will see called administration. In the perfect world, pills are the easiest thing because the patient doesn't know how to set up an IV. They typically don't know how to inject themselves. And I don't want to tell you about the other routes yet. Get there. Another one though. What in your body destroys chemicals. Start with pills. Where do they go right away? Stomach. Is the stomach a friendly environment or is it nasty? How much of that drug do you think is going to get out of that violent pH mess? Not 100%, that's for sure. So, stomach's going to take down some, it's going to take its cut, the liver's going to take a cut, and the kidneys will be filtering stuff out. Plenty of pills that we take that are commonplace drugs, you are only receiving actually like 4% of that. It can't be zero, it can't really be like 1%. We'll see this. I'm gonna ruin this. Bioavailability is the complicated, fun way to say that. So, assuming that we've designed a drug and we're going to break bad, it's good because it binds. It has a reaction that we can rely on in most patients. We can get it to patients pretty easily, and it works when it's not an IV. IV is 100% administration. It is the perfect route. It is not feasible clinically. Definitely not over the counter. Couple last ones here. You cannot have a drug if it has terrible effects, even if it accomplishes its treatment goal. If you kill the patient before you solve the disease, you're not going to market that drug, no matter how good it is at canceling out plenty of those things. Don't kill patients. This is the ultimate challenge with cancer medicine, because those are our cells. Most of what we're going to see are our cells, our heart cells, our brain cells. So you got to be very careful. You do not kill or cause damage. I don't want to poo-poo different drugs and stuff, but plenty of ancillary things out there exist where it's like if somebody starts anti-anxiety or antidepressant, the stuff that we'll see week 4, they don't go back to how they were. And that is a potential consequence. And it's not something you call a symptom strictly, but still. Okay, last one. Why does it have to be so small? It's got to be small. Drugs have to be small. You know how tiny H2O is, but that can just fly in and out of the cell everywhere it wants, right? Talked about blood-brain barrier. That's even stricter than 500. If you go above 500, you start to get in your own way. Too plump of a drug is not going to fit into the right spots. Too bad. Now, this changes when you consider an antibody a drug, an actual gene that we're sending in, or insulin. Those break the rules. We're talking chemical ones here. So, those are our basic setups. Not too bad. Now for my fun chemistry slide. I know we were all so excited for this. Lipinski's rule of 5. Here we go. So back in the day before biologics, but kind of like when we were in the drug design organic chemistry era where we're like, let's just see how this goes. There were 5 things that ideally a working drug would hit. So, like we just talked about, we want it to be small physically. Gotta fit. Go over here though. Why would having too many polar side chains be a bad thing? Too many. 1 or 2 is not going to kill you. You got those polar charges. Are they allowed to cross membranes? No. Body's made of water, right? Guess what water carries off with it? See a drug. Can't design something that's too happy to just get urinated out, basically. I'm going to say too H2O favorable. Not going to work all the time. Next one. Crystal's got their organic chemistry kits. We get the little 3Ds moving around. Yeah, oh yeah, I kept that thing. I lost it. So why would having too many chiral centers be bad? You want this thing to fit in a single target, right? If you got a million chiral centers running around, aren't you going to lose the shape every time? Because it's spinning around. See? That's why Dr. Bethel put you through that. Too many chiral groups. Inferior. His rule is less than 10, please, basically. Otherwise, it's going to look way too weird. Okay, next. The drug cannot be overly acidic or overly basic. It'll get chewed up by the body. Your body's at a natural 7, 3 in the stomach. Getting outside of that range is a bad time. I'm going to say too metabolized. For chiral, I'm going to say too wild. It's like flip and flopped all over the place. All right. And then the little last guy. It cannot be too attracted to lipids and fats, or it'll just stay in the lipids. Won't actually get anywhere. It'll get stuck. Unacceptable. There we go. Nice. So, plenty of drugs out there. Accomplish all 5. Plenty of drugs we'll see. Sometimes only do 3 or so. It's more of a guideline, but where the chemical factors kind of roll in, but they also are breaching into why biology-wise, it's not going to work in a lot of cases. So, always fun to meet these 5. Again, a decent framework for an exam question. I've worked on it. I don't do the what is not a thing as many times anymore. I thought I was going to become more evil in my old age and tenure, but I got merciful somehow, and maybe that was a mistake. We'll see. We'll see what happens on the exam. Okay. So here's a good example. And it's on the drug list, so it's red text. There's aspirin. Here's your normal COX enzyme. It's responsible for making platelets. It's a big old gene. You go to DNA, you have DNA for this. There's a serine right there, an amino acid. It's very sensitive at that active site. Remember how many times we punished you guys with enzymes in 105 and all that? Now we're actually using them. Look at all that aspirin does. Loosens up your blood pressure because it stops the platelets from forming and you're less dense. Less little red blood cells and platelets. More free flow. Less headaches. See our little target in there? Nice little aspirin's going to go and bind that serine and kills the enzyme. Can't function anymore. Aspirin's only 180 Daltons or so. Tiny little guy. Now I can't go on my history tangent here, but salicylic acid, what aspirin comes from a tree bark. Locals would take it when they had a headache, and then people found out and were like, hey, I have headaches too. People would actually pay to not have their headaches. But it's as simple as that. Find the spot that the gene that the target needs. Bind it up. Slice and dice. Done. Not every drug is as clean as aspirin. We'll find that, but pretty good. So, fun little green text knowledge here. If you take an aspirin and then ibuprofen actually is targeting that COX-1 as well, and COX-2, but different story. The ibuprofen won't really work. The aspirin's plugging the spots. Doesn't make any sense to take that. You're just going to get a stomach ulcer instead. Just do one or the other. All right. So how do we actually determine this? We've seen clinical trials in the news. Yeah, we talked about COVID. Talk about that too. Here we go. Most of you might have seen this a couple times, so I'll maybe go a little faster, but here we go. Phase 1. You grab a bunch of healthy people, you pay them about $50, they take the drug, And we're just looking for toxicity. These are not people with the disease that we're treating. This is just your normal people. I'm going to put a little note here. Sorry. No disease. Your toxicity. Is this going to actually kill people? Is it going to cause symptoms? Is it worth even investing anything in? Oh, I hate to do this, but there's a little guy up here. Look what I do. Preclinical. There's a whole phase up here. Sorry. That's like the lab and stuff. We're going to test it on the human cells like I do, or the mice. Call it phase 0 if you really want. Kind of a cool way to put it. So again, always a hard distinction on phase 1. We are just looking if this drug hurts people, normal people. If it does, the journey ends. Maybe it worked in the mice and the cells, but we aren't doing this, and that's okay. Now, phase 2 is when we start making some actual investigations. We grab a chunk of people, maybe, let's say 50 to 500, a small number, and they have the disease we want to treat. And we say, you take this drug instead. We'll compare how they do compared to the historical people, the ones that have already gone through it, died, whatever. Now, a true trial where you give somebody a sugar pill and somebody the drug, luckily, we don't have to always do that anymore. You can rely on historical data in most cases. But phase 2, this is a true small study. And we want to see in a survival curve or a response curve, did we make a meaningful difference? Was it better than the old treatment? Three is simply the same as 2, but massive. Thousands of people across the country. Different groups, different disease stages, everything. Different ages, everything we can do. We're going big study over here. All right. And last one, phase 4. Neat thing with phase 4, the drug is approved. The drug is being sold. The drug is good. It's just following up on everybody that takes it. It's just extra data. We're just being safe. That's it. So, if any of you have ever had the yellow fever vaccine, for example, that's technically in phase 4. We're good. You just watch it all the time. It's okay. Hey, don't worry, I had it and I haven't got crazy, so we're good. World Traveler, there you go. So 4, you just keep getting data. Eventually 4 ends, and you're good. 4 ended for aspirin, Tylenol, ibuprofen a long time ago. Plenty of cancer drugs. It's over. We're good. Data's data. So, given this, I think I have the question a bit down here. But the question often happens, why? Oh, shoot. Well, no, I actually already do this, so I'm not going to spoil it. Wait. Never mind. Yeah, I'm not going to spoil it. So why do we have this nightmarish admin? Why does the FDA slow all of our progress? Because those trials took years. And that's not counting the preclinical work. And a patent only lasts 10 years. So why are we so fussy? Why are there so many regulations? Who's ever heard of thalidomide? Yeah. So it was this was back in the day when women were just hysterical, so we just gave them this drug to calm them down because they didn't have actual symptoms, because that was about 70 years ago. So we moved on from that, luckily. Take this thalidomide, you're hysterical. Turns out the thalidomide didn't have as many symptoms in the actual person, but if she was pregnant, that thing went and crossed that fetal barrier and did all kinds of developmental nasty work. One person in an office in the FDA was like, there's way too many birth defects with these people on this drug than there are normally. And this goes deeper. Like I would definitely go into her story. She got like, you know, all the conspiracies about like people following you and all that stuff. The thalidomide people did all that. Followed her car, marked her. It was bad, man. Because it was a good selling drug. Everybody was taking it. Ooh. So this is the key thing. And I know this is a little bit of red text maybe that's a little lawyery. The proof that it's good is on them. Every drug is guilty until proven innocent. Proven innocent. Verse of our justice system. You have to have overwhelming evidence that you're fine. Because if it gets to that phase, that's a lot of kids that were born. I'm not going to show you the pictures. You can Google it during the break. Speaking of, long live the king. Ha! We're going to do some good Photoshops. This is my best Photoshop. There we go. All right. So we've breached into our Wednesday class, haven't we? We only have one more. Done pretty well. All right. 5 more minutes and you feel all right? Sounds good. I like breaking it up like this. Otherwise, I'd talk forever.
It's in the lab where I stand.
Yeah, exactly.
Oh, one little quick informal question. Is this the first time everybody's been in a 3-hour class?
Mhm.
Okay.
Well, one where you have to sit, like labs. You just
Oh yeah, that's true. Yeah, I promise we'll do our best. Don't worry. I have activities planned.
This is 2 hours. 15 minutes. Trying to calculate how much time we have left. Probably an hour exactly.
So my little advice slides, some of you guys like these. They get voted pretty high because students have a lot of questions for professors a lot of the time. I turned those into slides. Some of you guys know this one. I have some stuff that neither of us like, don't I? No answer key. Yeah, that one's tough. I don't particularly like that one either, but all the cognitive and psych people say, put a straight answer key out there with the questions. There's nothing that stops that temptation from being like, of course I knew that. That's the best of us. This means that I will do maybe some sort of scavenger hunt this year. I don't know. Maybe I'll like leave like answers around the building. I don't know. We'll figure something out. I bet there's a middle ground there. And then this is more for lab stuff. But I have a million people that are like, what do I do next? What do I do next? I can't do that. You have to do it. It would be quicker for me if I just told you what to do, right? Now, specific questions and such, yeah, fight me. I want to help. My instinct. I actually have to stop. All right. And then the last one. I will say this class does cater, as it's sort of a med designed class. Some of the content is very strict, like drugs. It does cater to quite a bit of the multiple choice. That's fine. I will try and do some longer differentiation answers. They're intimidating at first, but I do find that people do quite well on them. Like in cancer bio, one of our second exam, the median was an 86. And I was like, how offensive. It was way too high. Just kidding. I was proud of you guys. It was good. All right. Big one in this one is that something called Bloom's taxonomy. There's a pyramid. The easiest thing to learn is how to memorize stuff. The next thing is I can explain what that is. The third one, which is what I want to get to, I want to apply stuff. A good application question for this would be, hey, this patient's taking aspirin. What drug should they not take with that? What will interfere the most with it? That's a good way to apply something. So we'll see those once in a while. All right. The last section's fun. We get to rag on a bunch of, oh, wait, I gotta be careful because this is actually a little hot right now. I'm not gonna lie, yeah. So again, I'm a little animated. This is obviously for other purposes. And again, I'm not here to like brainwash people and be like, the world's evil and blah, and like go crazy, okay? Let's look at this. Let's see what we're dealing with. Let's see why the partnerships that we currently have exist, okay? Consider to yourself just as an open question. Your major questions about insurance, and trust me, you guys are graduating soon. You're gonna get kicked off your parents' insurance at 26, huh? It's coming. We don't like that. We didn't like that in cancer bio when we said that. It's coming, don't worry. Here we go. So let's do some life prep, huh? And if you want to work in healthcare, hey, like 30% of that's paperwork, man. Let's do this. All right. Here we go. So to be insured against massive costs, the kind of CT scans, bone breaking, drugs, anesthesias, this is what you do. You bet on yourself that that won't happen, but if it does, rainy day. Per month is the premium. That's the bill that you are paying every month. And if everything goes fine and nothing else happens, you just pay that every month, you're done. Deductible. This is what you're going to pay if something goes bad. You're going to pay that raw amount, about that amount. Notice the fashions of plan A versus plan B. Do you start seeing those kind of develop right now? The differences? If everything goes fine and nothing happens, who's the better plan? Might be, right? It's cheap. $200 a month. But man, you better hope you don't get in trouble. You get in that car wreck, right? And it's a couple leg fixes, a lot of anesthesia, a lot of drugs. They're going to run up at $5 grand. You'll meet the deductible. After that, the insurance begins to kick in, pending denials and approvals and et cetera. Coinsurance is a weird one, though. People will actually still get bills after you reach the deductible, and everybody gets mad. Coinsurance is that reason. It's down. You will still pay that amount after you reach the deductible. So there's more. Shoot. But there is a finale. There is a stat called out of pocket. That's the end game. Theoretically, that is the end game. That is the most you will pay, max. Coinsurance, deductible combined. So car wreck that runs you 100 grand, both these plans are going over and you will meet both out of pockets. So there it is. But take a look at the features of each. Which one of these is obviously more risky? Yeah, being kind of betting on yourself there. Insurance premiums will always be cheaper if you are a non-smoker and you're younger. Now, the pre-existing condition thing, that's a new thing. We used to be able to say, you have diabetes 1, your insurance is the roof, man. And it was free market. Kind of. One last factor. Hospitals are going through the business MBA transition right now, whether that's healthy or not. They work with the insurance companies, and they're either in network through contracts or they're not. If you're out of network and you get treated somewhere, you will pay that amount. Your insurance is not coming to save you because they didn't handshake with that place. So if you get in a car wreck, do it in the county you're from. So of all the stats that we just saw above, out of network will trump those. So if you have a nice insurance plan, everything looks good, but it's like of the 13 medical centers in this area that I'm in, only 2 are in network or 0, you can't take that plan. Yeah. Problem is, and why we're going through this paperwork stuff is patients will often need your help on this. So having these basics are here. Also doesn't hurt as we're all kind of going, eventually you're gonna have to buy this yourself probably. So everybody always wants single payer, huh? One payer, right? The idea is that that's the government, but one of my insurance friends explained something to me. We're in a single payer zone right now, one of the only ones in the country, because the only hospital in southern Minnesota that's in network, big guy, Mayo. All the little clinics that you see around here, the little Fairfields, some of those other little guys, they immediately have to reference you to the Mayo satellite. So your insurance kicks in. That is all they can do in a lot of cases. They technically are single payer right now because all the plans pay right to Mayo right now and they pay right back, and there's a mutual back and forth right now. Kind of interesting. So talked a little bit about trials, but I really wanted to check in on one right here, E1. So this is all the stuff. Remember that preclinical, that phase 0 that I talked about? This is the beginning of that. There's 4 stages to that. This is a lot of that organic stuff where it's like, I want to screen, I want to make sure this works, I want to have a target. I'm going to do some animal stuff. Eventually we get here, and then we start the phases. So the reason that I'm starting in the very beginning is that we're going to get the law involved a little bit here. Patents. Once something is created, and that number 1, that chemical, there's a 10-year clock on things. And now, where all these rules with clinical trials come from, it's going to take a long time. And I know it's a lot, don't worry. There's some neat things in here. Do you know where the clinical trial rules actually come from, their genesis? It was the Nuremberg trials after the Nazis were defeated. That there must be, do anything for medicine, there better be a good reason you are doing this. Most of these are going to say, you better be fair to all the people you are doing this to. Because what the Nazis did was take a bunch of twins and say, you're getting this and you're getting nothing. Let's see what happens. It was the beginning of that. And it identified that there was no world crime against that. So the biggest one that I highlight here is that drugs, when they start, they need to have that value. As of right now today, if you started and said, I'm going to design a pill that does what Viagra does or does what Rogaine does, that would not get approved. Both of those drugs were heart drugs that failed. They were used for something else. But there is no reason to put patients through the potential dangers of a new drug if it's not valid. Strange system, right? Still interesting. Now, the main thing that I want from this red text slide, how long do you think it's going to take? Compile all the paperwork. Get all this stuff on your good candidate drugs. How many years? Because we started the clock at 0 when we started the chemistry. Here's the problem. This will set us up for why drugs are expensive. In a typical trial, it takes years. And by the end, when you're in phase 4 and you are finally making money on your product as a big pharma, you've only got 3 years to do it, don't you? So you got to get all the pennies out of that. Now, this brings up a good point about trials. This is the point that I was like, oh, I can't make that yet. This is a common thing that comes up with COVID vaccine, right? That was quick. Oh, a little scary quick, right? We've known about Ebola for a long time. Big deal why this one isn't. That we've never been able to set up a control arm of any kind. Somebody is infected with Ebola, we hit them with the 3 main treatments: plasma, the serum, the antivirals, and the vaccine. We don't know actually what works. We've never been able to control for that. We've never had a population of thousands that have it and thousands that don't, that were both given the vaccine. It's never been controlled. COVID, though. That trial had about 30,000 in each arm. Why did it take so quickly? It's because, remember the times when the curves were all rising up? All 30,000 in this arm got it almost all 30,000 in this arm got it eventually. And we had the data done. That was it. Most diseases don't travel that fast, but most diseases aren't COVID-19. That's why it happened fast, because the data erupted. We can't even do that with flu. Doesn't infect that fast. Nothing ever has. So, a good little aside. But this one, I will pause on this one a bit. You're talking to somebody who has made partnerships with the industry. We did at Mayo quite a bit with Celgene and Bristol Myers Squibb. What is their role? And I mean true pharma industries, the big guys, the Eli Lillys, the AstraZenecas. You don't have to shout it out. You just think to yourself. I do link the full seminar that I'm going to kind of hit the major points on here. Vince Rajkumar is doctor at Mayo, multiple myeloma cancer. Takes $0 from pharma, so we had plenty to say. A good seminar. They're live. Lucky. Okay. Let's see the way that this works. Some of this has changed in the last year, though. All right. I have never seen so much money thrown around. I go to conferences for cancer stuff. I've never seen the money these pharmas have, okay? I remember one time we went to some restaurant and I started to have a panic attack because people were ordering stuff and I was like, shit, price. Nah, it's on the card. You guys are clients. And I was like, oh. But I didn't go crazy because my cheapness wouldn't let me, but I was terrified. The amount of money that was just hit, I was like, the amount of money it takes to rent some of these conference spaces. Now, a couple things, why things have gotten so big. People take more drugs now. You can debate if that's good or a bad thing. The revenue is climbing like anything else. Here we go. These are the major arguments that major pharma is going to make. And like I said, that patent's going to expire in 10 years. You got a little window there to get that before a generic pops up. And you can argue against generics all you want, but eventually people are going to pay with their money, okay? But these ones, most pharmas make one drug, right? Is there any other real competition beyond that? Sensitive population too. If you need that drug, that rare disease drug, what are you going to do? Not pay it? Very sensitive population. People like living. But let's start with the big one. Anybody know anybody type 1 diabetic in their life? Yeah. Insulin is a human gene. We could make it in the lab right there. Insulin as a product is still as cheap as it comes. You can make barrels of it. But if you keep calling it a new product and keep adding one chemical group here, one here, and name it something new and patent it again, you're off to a new beginning. But then why wouldn't you use the Walmart one, the Costco one? What happens in between? You're willing to pay, number one. Customers in this case can't really help themselves. It's not their fault. You need the insulin. Number two, most drugs are made by one maker. They have all the keys to it. Making a generic's not simple. Man, when a generic fails, the whole thing goes down in flames. Patents. Plenty of legal arguments to delay that 10 years into more like 15 to 20. Fair enough. But we are focused on insulin right here. Insulin was free. There was no patent. Once you start adding one little group where you make it a different way, call it something different, now you can sell it again. So patents aren't going to save us. But here's the one that's hard. You got a cycle going of drugs that when you change them just a little, you make a new product and you charge more. Look at the advertisement on the right in New England Journal of Medicine, the top academic journal in the world. It is telling physicians, Why would you give your patients and hurt them with the old insulin or the generic insulin? You're hurting this child. It's up to the doctor to prescribe where it comes from. So that's the first part. Physicians are not evil people, okay? This is the system. We're going through it, okay? Go here. Like I said, generics aren't easy to make. Doesn't always work. I would joke about making it in the lab right there, but I would not dose that to anybody. Number 6, insulin specific. Again, there are 3 major companies, Eli Lilly, 2 more, that make insulin. And magically, every time one company rises, the others rise just ahead. There's no competition. Somebody's talking. There's no competition. We're breaking that rule. The last part. There is a lot of transit between making the drug and you getting it. Talk about cuts. We're going to take a little slice of that pie each time, okay? Here we go. But here's the strange thing, and this is a very odd concept. So first time I had to see this twice. Pharma will charge the middlemen the top price. Middlemen will charge an even higher price each time, down to the patient getting the highest price. How's that possible? This is an oversimplification. But the system is designed by the drug company to say, oh, look, thank you for buying our drug. Thank you for prescribing our drug. Here's what we have. It's called a rebate. You can give a rebate to the wholesaler, give a rebate to the pharmacy that paid that full price, but not anymore. You give them a rebate, right? Give a rebate to the physician. Lastly, you can buy off, theoretically, is the way it's sometimes put, the patient. There are rebates for certain drugs that the patient will then get, so they don't feel as impacted. But who made the most money in this transaction? All the way at the top, even with the rebates. That's why, and I'm not trying to, we're not going to debate policy here, but this is who gets demolished. Zach, you're in a pharmacy. Do you have people that come in that are, I'm uninsured, but I'm going to pay for that? Yeah. And that's where things get rough. That's where a lot of that money comes down, bleeds. Where do you think all that credit card debt comes from in America? You're not going to die, right? You're going to pay your insulin. And if you don't know, you can get it somewhere else. So red text, but same idea. Sorry, we're getting a delay on here. We'll fix that up. Here we come with in and out of network again. Eventually, some of the plans will simply not cover the generics anymore. Oops, sucks. And now you will pay the full price for the generic without the rebate. See how it's getting complicated? It's meant to confuse you. Now, the key part of his presentation was, though, even though the middlemen seem like they're the devil, they're really sort of in this system that can't be fixed at this point. Are they going to take a bit of a cut? Sure, you can see that on the pie graph, but it's not some evil amount. It's not some thing. It's kind of business as usual, to be honest. Not a ton that these are actually pulling. Not as much as at least they can usually be blamed for. Okay. Last part, this is my favorite. Bam. Oh, hell yeah. Best lawyers on the planet. Here's the deal. The way we have Congress set up, you got to run for re-election every 2 years as a rep, right? You start your next campaign probably 6 months in, or with a year and a half left to go. You need money. That's that. That's what we got. And man, money can show up. Sorry, that was a little way too loud. So we keep getting more. We'll keep going bigger. The good news, I don't think I have this slide. Good news that a new thing, and so I took this slide out. Why is Medicare cheaper? Why is the VA cheaper? Why do veterans not get just priced to death on these drugs? Why do older people not see the same issues as uninsured people that are under 65? Medicare, VA, they can negotiate with the pharmas. They can take them, bring them to the table and say, no, get away. We're only doing this price. Best we can do. Or they'll go to the generic. I think a year ago, there was no way to negotiate. Now there is. But now every drug should be able to begin that negotiation process. That's a healthy step, I'd say. That's probably going to help a little bit. So, and at the end of the day, as a patient, how have you ever felt in that room? Do you feel like you're in control? Hell no. It's terrifying. Whether you're at the dentist, you're at the hospital, anywhere. You're trusting these people. And again, they're not evil. Here it comes though. If I'm a physician, there is a certain amount of money that I can be consulted on, or I get a rebate on if I prescribe this drug. That number is higher. In many cases, if the drug price is higher overall. Don't gaslight physicians, gaslight all the people that became admin that don't do anything. Sorry, that's in the recording. You can take me to court for that. But they're just doing their damn job. They had about $300,000 in debt. They hadn't worked for 5 years because they were a resident. What would you do? Take $50 or not off the ground? If you did, it probably wasn't going to hurt too many people. But there are a lot of physicians that take zero. And that adds up. Raj Kumar was one. That's why he could give this seminar. So if you go to the big medical conferences, there is a section that says conflicts of interest. You have to list who you get funding from. Guys, I've seen novels of where that money's coming from. And it can get as abstract as saying, honorarium, Eli Lilly, gave a talk. I don't know, that's it. Research funding, big deal. And in a lot of cases, I'm going to give them their credit right here. We would not be winning this fight right now in medicine without that sort of powerhouse that pharma is. There is not enough public money in the world to push through the amount of products we've done. So I get it. Think of it as in World War II, the Soviets. Stalin was not a good dude. But there were other bad things happening. So we wanted to live, right? That's the idea. So is there changes that need to happen? Yes. Are there really excellent pharma teams? Yeah. Bristol Myers Squibb and the Celgene team, they were the most scrutinized. They were the most scrutinizing to my project with their drug, R2-CHOP. They absolutely were like, is it working? Is it working? Are you sure? Are you sure? There are conversations that go the different way that say, are you sure we need to pull those older patients to the trial? Couldn't we just do the trial with the younger ones and show how good they did? That can happen in a second, okay? And I'm not saying go conspiracy time, guys. So, how was our fun little whirlwind tour of medicine, insurance, drugs, da-da-da-da? Hey, we did pretty well. Hey, dang, we're kind of early. Wow, don't speak too soon. Oh, woe is you.